everyone. I think all are going very good in this pandemic situation. So many of the people are going to the diagnosis tests like PCR test, rapid test and the serology test, and most of the people are not aware of the PCR test and the rapid test, actually what is going on in the test and what is the process involved in it, and how the PCR and the rapid test is helpful during this pandemic disease. So today we, the students of the RBVRR from the micro department, just came up with some kind of information to let you know about the PCR test and the rapid test and what are the differences between the PCR and the rapid test which gonna helpful for us during in this pandemic. So let us move on to the PCR test. As myself Sharma, I just wanted to let you know about the PCR test. So just move on.
How does the RT-PCR test of coronavirus is done? There was a lot of curiosity among the people regarding the testing of coronavirus. So today we will let you know about the most accepted test of RT-PCR known as real-time reverse transcription polymerase chain reaction. In the first stage, the swab is collected from the throat of the suspect. The sample is put by the special chemical which isolate the cell from the virus. Now the viral RNA is extracted, then the extracted RNA is further converted into the DNA, and after which the new DNA is treated as a cDNA or the complementary DNA. In the second stage, the multiple duplication of DNA are made using the technique called polymerase chain reaction, that is PCR. Now in the third step, if the cDNA matches with the SARS-CoV-2, then we can say that the coronavirus is confirmed in the person.
So what is PCR? Let us see.
Living cells have the genetic material. The genetic material is a set of instructions that how cell need to work and what function it will perform and how it will grow and eventually die. These are of two types, that is ribonucleic acids called as RNA, the deoxyribonucleic acids called as DNA. When we move on to the DNA, it is a double-stranded molecule that is found in all the organisms like animals, plants and some viruses. The genetic code is present in it and acts like a blueprint of that organism. RNA is a single-stranded molecule and does some functions when we compare to the DNA. PCR, polymerase chain reaction method, is a widely used method in molecular biology to rapidly to make million to billion copies of the DNA sample. Generally, our scientists take the DNA sample and amplify it to larger number of amount to study in detail. The PCR was invented in 1985 by American biochemist. Now, some viruses such as coronavirus can only contain RNA. They rely and penetrate into the healthy cells to multiply and to survive. Once inside the cell, the virus RNA take control of the cell and make it into virus making factory which produces more cells which are infected with the viral RNA, and these cycles goes on. So this is a present problem for a traditional PCR, because in traditional PCR only the DNA can be detected or copied. So in order, our scientists need to convert the viral RNA into DNA, and this process is known as reverse transcription. The copying of DNA to RNA is known as transcription, and the process which we convert the RNA to DNA is known as reverse transcription. Hence, using this technique of reverse transcriptase with the technique of polymerase chain reaction is known as RT-PCR. So the enzyme is used by a scientist to convert the RNA to DNA, and this PCR make it several copies of DNA in very no time.
Now let us see how the PCR works.
So when we convert the RNA to DNA using reverse transcriptase enzyme, the resulting DNA is called complementary DNA or the cDNA, because its sequence is complementary to that original RNA strand. So At this stage, we need to know that DNA copies are collected that represent all the RNA is present in original sample. So these RNA can include from both the virus or the bacteria, or it may be from the patient own cells. So we need to determine that whether the SARS-CoV is present in the sample or not. Generally, we know that the PCR helps in the amplification of the DNA into more number of copies. PCR involves in 3 basic steps which repeated up to 40 times. Coming to the first step called denaturation, all the double stranded DNA molecules are denatured. It means the two strands get separated at this stage. In the second stage called annealing, in this stage the pair of primers which are specific to SARS-CoV attached to each DNA strand. The primers are the short nucleotide sequence that complementary to unique sequence in the viral cDNA. So the specificity of the primers ensures that they bind to the only viral cDNA and not to the other cDNA present in the sample. Coming to the third step, elongation, the enzyme known as polymerase adds the nucleotides to the end of the primers using the original DNA strand as a template. Now it becomes complete DNA molecule with two strands. There are 3 steps: denaturation, annealing and elongation, repeating with the amount of DNA doubling at each and every cycle. If you start with one cDNA cycle, after 34 seconds you would have over 34 billion identical molecules. Now, in RT-PCR, a probe is added which gives the fluorescence whenever a new DNA molecule is formed. The increase of viral cDNA can be known by tracking the fluorescence signal. When the level of fluorescence exceeds the certain threshold, we can confidently say that the signal is significantly the background level. The CT, the cycle threshold value, is number of cycles required for the fluorescence signal to exceed the specific threshold value. The lower CT value present in the original sample indicating the higher viral load. The SARS-CoV less than 40 is confirmed as positive. It indicates that patients have the viral load and it is very infectious. The several studies have shown that viral load as measured by the CT value that can help in the predict the disease and the progress.
Now we have come to know what is PCR. So now let us see that how this PCR technique is used in pandemic, that is COVID-19.
Copies are made using the sequence of DNA template even down to a single DNA molecule. The PCR amplification means the detection and identification of the target DNA can be made in visual techniques based on the size of the cDNA. Generally, the PCR theory is same everywhere. As we said that the primer should be added or annealed to the particular region of interest. During the PCR, the first stage, the double-stranded DNA is heated and gets separated, and upon the cooling, the primers get bind to the cDNA and also processed by adding the nucleotide by the polymerase and can be begun. So the initial stage, heating step, is called denaturation, is up to 94 to 95 degrees centigrade, and it needs the temperature at least long enough so that it can denaturate it completely. So now the following step, when the primer is annealed or attached to the cDNA template at lower temperature, is known as annealing, and the temperature is dependent on the specific primers. It is usually up to 50 to 65 degrees centigrade, and it does not need very long time, and generally we do use the TAQ primers in this technique. At the final stage in this cycle, the extension is up to 72 degrees centigrade. This is the optimum temperature of DNA polymerase to build the complementary strand. So the duration of this step depended on the length of the DNA, but Usually 30 seconds to 1 minute is most long enough. So this processing of denaturation, annealing, elongation is repeated to 30 to 40 times. Theoretically, doubling the DNA copies at each cycle and then by increasing exponentially the number of copies of targeted DNA. The sample can be prepared and kept in the incubator, and the complete PCR can be performed in the few hours or less than an hour.
In the previous part, we have learnt about RT-PCR testing technique. As there is an increase in large number of COVID cases, it's very hard to perform this experiment in a large scale. So most of the countries are relying on rapid test technique for COVID-19, which provides us a results in only 15 minutes. The presence of the antibodies pertaining to that of the coronavirus helps one to detect easily. The presence of the specific antibodies, that is IgG and IgM, helps us to know whether a person is infected with coronavirus or not. So, before performing the test, we need to know about the two very important antibodies, that is IgG and IgM. IgG, it is the most common antibody and it is seen in blood and other body fluids. It protects us against the bacterial and viral infections. IgM, it is found mainly in blood and lymph fluids. This is a first formed antibody when one is exposed to the new infections. As this is a very easy technique, we can perform this COVID test at home by an individual. Now, let's see how we can perform this COVID test with the help of a rapid test kit. Now, open the pouch, take out the IgM or IgG cascades. Place the kit onto the table as shown in the figure. Now add 10 ml of serum or plasma sample or 20 ml of whole blood sample into the test well, as is shown in the figure. Add 2 drops of the sample diluent, which is given along with the test kit. Now wait for 15 minutes to know the result or to determine it. Interpretation of the results. If there is an appearance of C line and T line, the person is tested positive, and if there is an appearance of only C line, the person is tested negative. If there is no appearance of any line, the test is considered as an invalid test. So by following these 4 steps, one can detect if a person is tested positive or a negative. If a person is tested positive, the proper medications should be taken. If a person is tested negative, he should take the proper precautions regarding it.
As you've heard about RT-PCR test and rapid test, let's see a few differences between them. In case of RT-PCR, it is used to identify pathogen genome like DNA and RNA, whereas in the case of rapid test, it is used to identify antibodies like immunoglobulin G and immunoglobulin M. In case of RT-PCR, the sample required is nasal swab or throat swab, whereas in the case of rapid test, the sample required is blood or plasma. RT-PCR test includes steps like denaturation, annealing and extension, whereas in the case of rapid test, it is a one-step process. In case of RT-PCR test, lysis buffer is used as a reagent, whereas in the case of rapid test, solution buffer is used as a reagent. In case of RT-PCR test, it can take 48 hours to few days to get test result, whereas in the case of rapid test, it can approximately take 30 minutes to 1 hour to get test result. RT-PCR test kit is large and expensive, whereas rapid test kit is small and inexpensive.